Okay, so welcome everyone to the NPTEL course on post-harvest operations and processing of fruits, vegetables, spices, and plantation crop products. So this course is instructed by Professor H.N. Mishra sir, who is a professor in the Department of Agriculture and Food Engineering, IIT Kharagpur, and I am Shikhar Sharma. I am a research scholar in the Department of Chemical Engineering, IIT Guwahati. So I am the TA of this course for this July to October, or we can say November 2026 semester. So this is basically a week 5 live interaction session, and the assignment deadline for week 5 is basically 26th of August. So please submit your assignment on time. I hope you have all submitted your previous 4 assignments on time. So this is the assignment 5, deadline 26th. So please submit on time. So the topics that we will be going to discuss in the today's class is basically including the module 5. So this module 5, basically, it is including topics like minimal processing strategies, hurdle technology concepts, then intermediate and high moisture fruit products, and then cut fruits and vegetables. So it includes basically lecture from 21 to 25. So 21 lecture is mostly focused on minimal processing. Then lecture 22 is basically focused on hurdle technology concept, and lecture 23 is about intermediate and high moisture fruit products. And the last two lectures, lecture 24 and 25, is mostly dedicated to the cut fruits and vegetables. So I hope you have gone through all these lecture 5 videos from H.N. Mishra sir's lecture. So now basically we'll be in this live interaction session of week 5, we will be basically solving some of the numericals, and we will be discussing some of the concepts regarding these 5 lecture videos. And so basically, if you have any doubt also regarding the previous any like week 1 to week 5 till week 5 lecture videos, you can ask. And if you have any questions which is unsolved regarding week 1 to 5, you can also ask those questions. You can ask these questions either in the live interaction session, or you can post your queries on Q&A discussion forum so that your queries can be resolved. And please try to study as much as possible, and please be with the pace, because we are in the week 5. So please cover all the lecture videos until 5 module, so that it will be easy for you to cover this course if you will be with us. Okay, so let's start this class. So this class, the first question is basically the concept-based question for this class. Okay, which of the following statement are incorrect regarding minimally processed foods? So there are 4 statements basically about minimally processed food products. So we need to tell which one is the incorrect one here, the incorrect one. So first statement is saying that they undergo a mild preservation process. Second is their processing temperatures are always above 100 degrees Celsius. Then statement 3 is saying that they require refrigerated storage and distribution. And fourth is saying that they usually have water activity which is below 0.5 and pH which is lower than 4.5. So we need to tell which statement is incorrect here. Okay, so let's see the answer. So statement 2 and 4 are basically here incorrect. So let's first understand what is minimal processing. So minimal processing is when the fresh produce, it is subjected to different mild processing steps to obtain a fully edible and natural product while providing the convenience and functionality to the consumers and ensuring the food safety. So minimal processing means that we are basically processing any food product or fruits or vegetables like in such a condition that it is processed minimally and we can the consumers can eat like it's a ready to eat food product. Simultaneously, it should be as close as to the fresh produce. So it should be damage, its quality damage is minimal and it is similar to the fresh like produce. So least possible treatment is basically applied to these kind of food products so that the defined purpose is achieved and the influence of the quality characteristic in the of the food product will also be going to be minimal. So maybe suppose we have the purpose of achieving that the product should not have this kind of pathogens or it omits some kind of eliminates or omits some kind of the pathogens from it. So also the purpose is that the food product will not have those particular pathogens or spoilage causing microorganisms. So, along with the processing step which we are applying, it should minimally damage the quality characteristics of the food product. So the food product will be in the as fresh as close as fresh like product. So at the same time, giving the food sufficient shelf life during storage and distribution. So, in the minimally processed food products, what we are going to do, the minimal or the least possible treatment will apply so that the defined purpose of killing of the microorganism of the pathogen will also be done, and simultaneously the quality characteristics of the food product can also be maintained. And since uh, sometimes we can have some defined purpose that the uh, quality characteristics should be maintained along with that the food product should have a sufficient shelf life so that it suppose 1 month shelf life or 2 month shelf life, so it can reach to the consumers in a good state and also it is the fresh like produce, so that is the more in demand by the consumers. So the minimally processed food, they basically undergo mild preservation process, then product temperatures. During processing, it falls between 0 to 100 and it will not go beyond 100 because beyond 100, the quality of the food product may alter. So it will be from 0 to 100. So first statement is correct that they are basically in the minimally processed food products, mild preservation processes are basically applied. And the statement second is incorrect because it is saying above 100 degrees Celsius processing temperatures, but not less than 100 degrees Celsius processing temperatures we keep generally because we want to apply mild preservation techniques. Then rely on refrigerated storage and distribution. So basically in minimally processed food products, there can be multiple hurdles. Like we can apply heat and then we can store that the food product in the refrigerated storage condition and we can change the water activity and we can change the pH so that all these preservation techniques will be applied in a mild level so that the product quality will not hamper that much and we can achieve uh, desired shelf life and killing of the desired level of the microorganisms. So it relies basically on the refrigerated storage and the distribution. So which is correct, statement 3. And then They have the water activity which is greater than 0.85 and the pH 4.5, greater than 4.5. So statement fourth is incorrect here, that water activity below 0.5 and pH lower than 4.5. So statement 2 and statement 4, they are incorrect, and 1 and 3 are correct here. So according to the NOVA group, so there are the different classification of the food groups. So the food groups can be classified basically in the different classes like unprocessed or minimally processed food products, then the processed culinary ingredients, and then processed food products, and then ultra processed food products. So we have these different kind of food products category. So if we see here, suppose we have this pineapple, fresh pineapple product. So this fresh pineapple is basically when it is this is a basically fresh product. So when it is converted to the cut pineapple, it is a minimally processed food product. And when it is basically put in the syrup and put in the can, it is a processed food product. And when the jelly or jam from this pineapple is being made, it is called the ultra processed food product. Like this is a fresh corn. So this is a fresh product. And when this corn is basically, we can take out all these uh, kernels of the corn. So that is a minimally processed corn. And then if we basically, we are putting it into some can and we are processing, that is a processed food. And when this from this corn, if some kind of snacks are being made, like uh, these Mad Angles, Doritos, like these chips or these kind of products we are making, that is the ultra processed food product from the corn. So these are the different classifications from of these food products. So as we discussed that the group 1 includes the unprocessed or minimally processed food products. So these food are in their basically natural state or have undergone minimal processing operations before consumption. Example like the whole muscle meat, seafoods, egg, milk, fruits, vegetables, and nuts. Group 2 is processed culinary ingredients. So it means that these food ingredients basically are extracted from the natural substances and only undergo simple traditional processing methods, such as the pressing or grinding or milling or drying and refining. Examples includes like the beef tallow, then lard, butter, salt, sugar, and vegetable oils. Then third group is processed food. So these food products basically are processed and they are typically produced by combining group 1 food with the group 2, which is processed culinary ingredients. So when we combine this group 1 with group 2, we can get this category 3 products. So these group 1 and group 2 products will be combined using simple preparation methods such as the salting, pickling, smoking, canning, or fermentation. So that includes group 3, which is processed food products. So it includes basically salted, pickled, cured, or smoked meat and fish products. Then processed food contain relatively few ingredients and are typically prepared in home or restaurants or kitchens. So in group 4 is the ultra processed food products. So these are the food products which are highly processed products and they are made using industrial techniques and often contain numerous additives such as the preservatives and the colorings, flavorings, and emulsifiers. Example includes like bacon, chicken nuggets, then ready to eat prepared meat products, then reconstituted meat products. So the industrial processes basically are used to make these products such as like extrusion or centrifugation or ultra filtration and high pressure processing or high pressure homogenization. So they are not typically available in the home or restaurant kitchens. So these are the basically made in the industries. So these products are the ultra processed food products. Okay, so in the previous question for the minimally processed food product, that is the we are keeping the food product in the minimal processed state. So statements 2 and 4 are false and statement 1 and 3 are true. Okay, let's discuss this next question, which is assertion and reason type question about enzymatic browning process. So basically this, when we are having this minimally processed fruits and vegetables, so there can be different quality changes can be occurred in these minimally processed fruits and vegetables, like physiological changes can happen, then microbiological changes can happen, then nutritional level changes can happen. So in the physiological changes, this enzymatic browning comes. So here we are discussing about this enzymatic browning of fruits and vegetables basically caused by polyphenol oxidase. And reason is saying that the polyphenol oxidase, it catalyzes peroxidation and it forms false smell, aldehydes and ketones. So we need to tell if the assertion and the reason are true or false, and if reason is correctly explaining the assertion or not. Okay, let's see the answer. So here assertion is true, but the reason is false. So in the physiological changes, as we discussed, this enzymatic browning comes. So this enzymatic browning of fruits and vegetables, they are basically caused by polyphenol oxidase, which is the assertion and it is true. So here we can see in apple, this enzymatic browning is happening and the apple is converted into the brown color. So what is happening basically? So there are different reactions which are taking place in the basically apple. So this is the polyphenol oxidase substrate is there. And because of the enzyme polyphenol oxidase, it is converted into some product, and that is basically going to further convert into some other like oxidation is happening. So oxidation coupling is happening, and like basically some other product also it will convert, and these can be aldehydes or the ketones, or it can be any other compound which is going to form this brown color. So what is basically happening is, so due to the polyphenol oxidase, this hydroxylation and this oxidation reactions, they basically take place. So like monophenols, they are basically, this is the monophenol. So this is basically converted into diphenols using this polyphenol oxidase and in the presence of oxygen. And further in the presence of oxygen, this polyphenol oxidase, it converts diphenols into orthoquinones. So this is basically the product which is going to help in the browning and sometimes in the like uh, basically in the browning. So these basically compounds, they are formed and it is going to be changed, or they are basically oxidized. Some products are going to be reduced. So basically the oxidation process takes place, and in the presence of oxygen, this polyphenol oxidase will act and it will convert the certain products like the phenols into the quinones. So this is the basically uh, what is happening when this is the reaction basically. So during the enzymatic browning. So this is a simplified process of enzymatic browning and inhibition mechanism of anti-browning agents. So the process of enzymatic browning, it initiates with mono phenolase activity from a paraphenolic compound to a 3,4 polyphenol. Then after that, enzymatic polyphenol oxidase activity will be taken place. So it will to produce orthoquinones derivative from 3,4 polyphenol compounds, and then those orthoquinones are basically the compounds which are giving the basically brown colors. So this is basically monophenols. So this is basically a colorless compound, and on this basically monophenolase will act in the presence of oxygen. So this mono phenols, it will convert into diphenols. So ortho diphenols is converted and it is a colorless compound. After that, basically this diphenolase activity will act. So it is basically uh, converting basically, or we can say polyphenol oxidase will act here, polyphenol oxidase here also and here also, right? So it will act in the presence of oxygen, and then it will convert diphenols into orthoquinones, which is a browning compounds. And when like there will be amino acids or protein, it will convert into complex brown polymers, which is also like having the color of brown. So in when we are saying that we are applying some antioxidants, so these antioxidants basically react with either with this compound or either with this compound, like the ascorbic acid or N-acetyl cysteine or 4-hexyl resocinol. So these are the basically compounds like unripe grapes, or the basically the antioxidant compounds from the different fruits or the vegetables. It will come like glutathione. So these are all the compounds which is going to come and act in this state, either ortho-diphenols or the orthoquinones. And some of the other kind of the compounds which will going to help reducing the browning includes like basically chelating agents. So they are basically like EDTA, kojic acid or carboxylic acid. So they will also help to interfere with the polyphenol oxidase activity, and some of the acidulants which will going to lower the pH so that the polyphenol oxidase will not act properly, like the ascorbic acid, malic acid, citric acid or phosphoric acids. And then there are different like other things like lime juice or pineapple juice or wine or onion solution. All these have this competitive inhibition. By the mechanism of competitive inhibition, so they will going to act on this polyphenol oxidase so that its activity can be inhibited, and those compounds like the monophenols or the polyphenols, they or the diphenols, or they cannot convert into this orthoquinones, and if they are not converting into orthoquinones by this polyphenol oxidase, further this brown color cannot be seen in the food products. Okay, so next is uh, so the first statement, which is about the assertion that the enzymatic browning of fruits and vegetables, they are caused by polyphenol oxidase. So as we discussed in detail, so it is true that the polyphenol oxidase is the one which is going to basically convert these uh, monophenols to diphenols to orthoquinones and give the browning. Okay, so next is the reason. So reason is saying that the polyphenol oxidase is the one who catalyzes this peroxidation, and then it forms the fouling smell, and it will create these aldehydes and the ketones which are going to contribute to the fouling smell. So basically lipooxidase and the also known as the lipooxygenase. So they are the basically one of the same enzyme. So they basically catalyze this peroxidation. Okay. And it will result in the formation of the falling smell aldehydes or the ketones, but not the polyphenol oxidase. So the reason is incorrect because the lipooxygenase enzymes are the one which is going to convert or which is going to convert these basically which will this organic acids or this uh, lipid compounds into the, like they'll oxidize these compounds and like the fatty acid compounds basically. So the fatty acid compounds basically they will be converted into aldehydes and the ketones, which will give the falling smell. So their oxidation is basically take place. We say that the fats when we store for a long period of time, it will oxidize and the rancid smell will start coming. So that is what is happening, that the fatty acids are going to oxidize and convert into aldehydes and the ketones, which are going to give this foul smell. And it is basically take place with the help of the enzymes like lipooxygenase. So here we can see that enzymatic pathway of the aldehyde formation from linoleic acid and also alpha linolenic acids. So here this is linoleic acid, right? It is basically and here is a alpha linolenic acid. When this LOX, which is the lipooxygenase enzyme, it is acting on this, they are further converted into, like the linoleic acid, it is converted into 13 hydroperoxides of linoleic acid. So in the peroxide form, it is converted. And then further this HPL, which is hydroperoxide lyase enzymes, will act, and this is converted into hexanal. So that is an aldehyde is formed, right? So also this alpha linolenic acid, it is basically when it is basically act by the lipooxygenase enzyme, it is converted into 13 hydroperoxides of this alpha linolenic acids, and then further this peroxide lyase enzymes will act, and it is converted into its hexanal form. So that is the aldehyde form, which is going to give this fall smell. So sometimes this linoleic acids, it can also convert into its 9 hydroperoxides form, and then it's into the AL, which is aldehyde form, nonenal. So it is basically going to convert into its aldehyde form. And this same with the alpha linoleic acids. So this is also converted into 9 hydroperoxides using this lipooxygenase enzyme. And further, when the hydroperoxide lyase enzyme will act, it will convert into nonenal, which is a aldehyde. Like this, alpha linoleic acid, when it is act with lipooxygenase, it is converting into 13 hydroperoxides, and after acting the hydroperoxide lyase, it is converting into hexanal. Then further, it is acted by this enzyme alkenyl oxygenase, which will convert it into this particular 3-hexanal compound into 2,4 hydroperoxy 2-hexanal, and then this next enzyme will act, which is peroxygenase, and the compound is further converted to 4-hydroxy 2-hexanal. So like this, there is a chain of the different compounds are forming when different enzymes are acting. But mostly these uh, reactions will start with the help of the lipoxygenase enzymes, and further different enzymes will act, and it is going to convert into it, this fatty acids will convert into its either the ketone form or the aldehyde. So here, from here, we can say that the reason is false here, right? So these are the some other different physiological changes which will happen uh, when we have this minimally processed food products and they are undergoing some quality changes. So this is all related to the physiological changes. So other physiological changes includes like there can be also increase in ethylene production, so which will lead to the physiological disorders of the sliced fruits, like the softening can also occur, and the respiration activity of the produce may increase by 20% as well to as much as 700%, depending on the produce or also the which cutting grades, how we are cutting, and then the instruments we are using for cutting, and also on the temperature. So this are all the other physiological changes which is happening. So when the minimally processed food products are with us, so mm, ethylene gas can also produce from these sliced fruit products, and because of the ethylene is producing, softening or other degradative reactions can take place. So this is basically the mechanism of uh, this when the ethylene is produced, what is happening. So it is in the tomato fruit ripening process at the cellular level is shown here in this diagram. So the ethylene is evolved. So ethylene, when it is produced, this is ethylene. So when it is produced, so what will happen? It will going to be involved in the color, aroma, and softening of the this tomato fruit. The climatic fruits, basically, this climatic fruits, which is tomato, apple, like these, they basically are characterized by an autocatalytic production of this ethylene and by respiratory crisis. Other signal molecules are also uh, in, involved fruit in this process. So it's basically in the climatic fruit, as we know that after the harvest, the ethylene gas is produced. So that is a autocatalysis process, and this ethylene, when it is produced, it produces some signals, and it will change. It will the signals will go at the transcriptance levels, and some kind of signals will produce like the for the like the genes will produce, gene expression will be there related to the ripening. So because of the ripening, cell wall degrades and softening process will occur, and also the membrane degradation takes place. And because of the ripening, the color producing compounds in the uh, like all the pigments, they are in the chromoplast, they can also generate some kind of aroma compounds, or the volatile compounds will also generate. Then more ethylene will also produce from this, and the other compounds which is like sugar, acids, they will also degrade or decompose, and the taste will also change, and respiration rate, which is in the like which is also going to increase because of the these all the ripening related changes. So they will occur, they will produce or they will generate when the ethylene gas is reduced due to the autocatalysis, and the ripening related all these changes will take place. Then, as we were discussing, so when the ripening related change is taking place, so respiration is also one of the process. So the respiration rate is basically it is telling about how much of the oxygen is consumed and how much is the CO2 is produced into the by a cell like a tomato fruit cell or any fruit cell or vegetable cell. So it this respiration rate is also basically increased 20% to as much as 700% based on the temperature or the which kind of produce is there. So if it's a climatic fruit or it's a non-climatic fruits and how we are cutting the fruit. So minimally processed fruits, they are basically sliced or they are basically cut. So they are producing more respiration rate as compared to the whole fruits. And that based on the temperature, so we can see here that the respiration rate it increased based on the temperature. And we also use one of the formula like Ventoff formula for calculating like how the respiration rate or any reaction rate is changing when there is a rate in temperature increases there. Other changes are like anaerobic packaging conditions lead to the anaerobic respirations, causing the formation of ethanol, ketones, and aldehydes. So because of the uh, the minimally processed fruits and the vegetables they are in the packaged form, so their uh, the fermentation, alcoholic fermentation, or basically lactic acid fermentation can take place, and these ethanols, ketones, and aldehydes can produced and can because of the fermentation. Uh, these glucose will be consumed, and suppose in the lactic acid fermentation is happening, so glucose will be converted into lactic acid and energy use, and the enzymes will act like lactate dehydrogenase, and in the alcoholic fermentation the glucose is converted into ethanol, carbon dioxide, and energy, and alcohol dehydrogenase enzymes will take place. So these are all the physiological changes like take place, like there can be uh, like this browning, enzymatic browning can be there, or there can be because of the this acting of the lipoxygenase enzyme, there can be produce of this aldehyde and the ketone compounds, which can result in the foul smell, and there can be increase in the ethylene, and because of that, all the ripening related changes may take place, and respiration rate can also increase, and also the fermentation can occur when there is a different physiological when there is a different physiological changes happening when we are storing this uh, minimally processed fruits and vegetables, and the quality degradation process is taking place. So in this question, basically, assertion is true and the reason is false, as we discussed in detail. Okay, let's discuss now the hurdle technology concept. So which of the following is are correct with respect to the hurdle technology? So we have four statements like preservative factors or the hurdles, they disturb basically the homeostasis of the microorganism. Then second is that the microorganisms are able to overcome all the hurdles present in a food product. And then third is hurdles prevent microbial multiplication and may lead to the inactivation or death. And then fourth statement is hurdles technology involves interaction of the multiple parameters like temperature, water activity, and the pH to ensure microbial stability. So we need to tell which statement is correct here. Okay, let's discuss. Okay. So we were discussing about hurdle technology. So let's discuss what is hurdle technology first. So it is basically this hurdle technology, also known as combined method technology. So it is basically the in hurdle technology, what is going to happen? An intelligent combination of various preservation techniques of the hurdles are basically applied. So the hurdles, the like there are a combination of intelligent hurdles, or basically there are a combination of different preservative techniques are applied in a hurdle technology. So the hurdles are chosen carefully in order to maximize the preservation or preserving effects while minimizing the impact on the product quality. Usually, this is achieved by combining processes with synergetic effects. These hurdles basically includes physical, chemical, biological factors that create an unfavorable environment for the microorganism to grow while maintaining the product integrity. So there will be different hurdles will be applied, like there are different hurdles like water activity, then pH, then temperature. There are different hurdles can be applied, right? So if microorganism is able to cross this hurdle, so it will not able to cross another hurdle, or if it's cross another hurdle also, so it will not cross third hurdle. So it will stuck here, so that the microorganism growth can be hampered. Like here in the image, one hurdle microorganism already crossed, but the another hurdle is not able to cross. So we can apply the multiple hurdles to uh, stop the growth of the microorganisms, or to decrease the growth of the microorganism. So the hurdles are applied basically to the product for the disturbance of the homeostasis of the microorganisms. So microorganisms, they basically are not able to jump over all these hurdles present in the food products. So the preservative factors so prevent basically microorganisms from multiplying and then causing them to remain inactive or even in the die state. So the hurdles, they can be classified like physical hurdles like high temperature or maybe low temperature, and then electromagnetic energies like the microwave or the radiation, and then packaging techniques like active packaging or the edible packaging, and then physio physiochemical factors can also be there, like the CO2, O2, ethanol level, and then lactic acid, low pH or low water activity, low redox potential, organic acids and phenols, then salting. So sorry. Then yeah, then the salting and then smoking, and then sodium nitrate or the sodium nitrite, and then sodium potassium sulfides and spices and the herbs. Then there are other methods like the non-thermal processing methods, which includes high uh, hydrostatic pressure and the pulse electric field, pulse light, and so on. And the last one is the microbiological factors like the competitive floras, protective cultures, and microbial products. So these basically are the different hurdles we can apply in a food product, like the physical, physiochemical, non-thermal, and microbiological hurdles. So here in this question, we said that 1, 3, and 4 are the correct one. So about hurdle technology. So let's see the one, which is preservative factors or the hurdles. It basically going to disturb the homeostasis of microorganisms, so which is true. And then the microorganisms are able to overcome all the hurdles present in a food product, which is false, because it should not overcome or jump over all the hurdles. So hurdles prevent microbial multiplication and may lead to inactivation or death of the microorganism, which is true. And then the hurdle technology, it involves interaction of the multiple parameters like temperature, water activity, and the pH to ensure microbial stability, which is true that we can apply a combination of the methods, the combination of the hurdles so that the microorganism will not able to thrive in that environment. Okay, so this is our next question. So the assertion here is saying that the uh, food hurdle technology food, it remains safe and stable even without the refrigeration. And the reason is saying that combination of multiple preservation factors in hurdle technology is generally not act synergistically for killing the microorganism. So we need to tell if the assertion is true and the reason is true or not also, and if reason is correctly explaining the assertion or not. Okay, so let's see. So here assertion is true and the reason is false. So already we discussed what is hurdle technology. So next is that we have the different advantages of the hurdle technology concept, that it can avoid the severity of one factor for the preservation. And combination of multiple preservation factors in the hurdle technology is generally going to act synergistically for killing the microorganism. So hurdle at lower concentration, it prevents undesired side effects and it lowers the production cost as well and saves the energy. So this hurdle technology basically, it is a combination of multiple preservation technology and which will going to act synergistically basically to kill the microorganism. So the reason is the statement of reason is false here because it is actually producing a synergistic effect. And these hurdles basically using the natural preservatives in combination with the synthetic preservatives. So here hurdles we can make as per our choice. We can include some of the natural preservative methods with the synthetic preservative methods. And these hurdle technology foods, it remains stable and safe even without the refrigeration. So, which is true, assertion is saying same. So it is true. And the hurdle technology targets basically the physiological aspects of the microorganisms and it will either kill the microbes or the pathogens or suppress their growth. So if there is any harmful microorganism, we can have different uh, combinations of the hurdles. So we can even have the gas, altered gas of the or the atmosphere, and we can have the different chemical preservatives and temperature we can apply, pH, water activity we can change. So that is going to all will going to act together and it will going to kill the microorganism because microorganisms they will not able to maintain their homeostasis one because one after another there are different hurdles. So uh, here we can see that the microorganisms they are suppose first is temperature we are applying. So microorganisms it is crossing the temperature hurdle. Then water activity is there. So that is microorganisms able to grow here also. Then we are applying pH. Microorganism can grow here also. Then we are applying redox potential. Then microorganism is able to grow here. Then we have some preservatives. So here microorganism is not able to cope up with this hurdle. So we can have multiple hurdles in combinations in the hurdle technology. So this hurdle technology, it involves four major mechanisms to kill or delay the growth of the microorganism. So first is basically homeostasis. So the homeostasis is basically the tendency of the microorganisms to maintain uniformity and stability in the internal microbial cell, which is essential for its survival. So microorganism, suppose this is a microorganism cell. So it will try to maintain the homeistic or the uniform or the stable condition so that it can grow with its environment, and its internal and its outside of environment conditions should be in homeostasis. So once the homeostasis causing microorganism disturb, so the once the homeostasis causing microorganism disturbance through the incorporation of any hurdles, so then the microbial spoilage will either be delayed or microorganism will be killed. So once there is a disturbance of this homeostasis, so microorganism will try to balance with the hemostasis, but then it will take some of the energy, and due to the multiple hurdles, the energy will be exhausted, and finally the microorganism growth will be delayed and may be killed. So the stress factors can be low level of the nutrients. So basically low level of the nutrient will going to have low level of the metabolism in the microbial cell, stationary phase, and the viable non-culturable forms. Then low pH will going to removal of the protons from the cells and maintenance of the pH gradients. Then low water activity. So microbial cell have these responses of these stress factors. So when there is a low water activity, osmoregulation is there, then accumulation of the solutes, reduction of water loss, maintenance of membrane turgor. Then low temperature, because of the low temperature, cold shock response, change in the membrane lipid composition to maintain the fluidity. Raising temperature, so heat shock response will be there, change in the membrane lipid composition and their enzyme. Then if we are raising the O2 level, enzyme protection or the production of the catalase, peroxidase, superoxidase, and dismutase from H2O2 and the oxygen derived free radicals. And if there is a presence of biocides, then phenotypic adaptation or the reduction of the cell permeability. And if there is this ionizing radiation, so there is a repair of the single strand breaks in the DNA. And if there is a competition, so aggregation of the cells to show symbiosis and produce biofilms. And second is the second mechanism is basically metabolic exhaustion or auto-sterilization. So in hurdle treated products, microorganisms used energies in the maintaining of their homeostasis. In the end, microbes will be metabolically exhausted and eventually die due to the starvation. So this process is known as the auto-sterilization. So in auto-sterilization, it provides foods against, so it provides food safety against the pathogens and the spoilers during the storage, mostly at the room temperature. So suppose we have this change in the pH or change in some, like here we are discussing that the pH is changing. So initially the pH was 5 to 7, and after that, if the pH is changing to 1 to 3. So when pH is 5 to 7, so the basically the internal membrane pH, the inside the cell or inside the internal membrane, the pH is basically 7 to 7.6 in a microorganism. And basically what will going to happen? This microorganism, it is trying to maintain its pH as per the surrounding atmosphere. So when the pH is decreased further to 1 to 3, the pH inside this cell is basically from 5 to 7. So it is also decreasing from 7 to 7.6. It is changing from 5 to 7. So it is trying, this microorganism is basically trying to maintain the homeostasis condition, and it is trying to maintain the pH condition according to its external environment. Next mechanism is stress reaction. So the stress reaction has been observed in microorganisms when they are under stress due to food shortage. So microbes become more resistant under the stress as they release the stress shock proteins. So the rate of formation of this stress shock proteins, it depends on pH, water activity, ethanol, and heat. The synthesis of these shock proteins, it becomes difficult when there are more hurdles are introduced at the same time, which is done as in the hurdle technology concept. So it leads to the metabolic exhaustion of the microorganisms. Then fourth is the multi-target preservation of the food. So the combination of the different hurdles like the pH, water activity, or redox potential, they will going to act synergistically on the cell of the microorganism. So the multiple hurdles, since they are applying simultaneously, so it targets different cellular components such as the cell membrane integrity, then some are targeting enzyme function, then DNA stability, pH, hemostasis, and then water activity levels. So the synergistic level of the hurdles, it disturbs the hemostasis of the microorganisms and it reduces the synthesis of the stress shock proteins. Simultaneous exposure of the microorganism to these different stresses will sharply reduce the energy of the microbial cell. So the synergistic effect of different preservative factors or the hurdles with small intensity is more effective on microorganisms than the one preservative factors of the larger intensity. So, and suppose if we are applying one preservative factor, and it is at the, suppose the temperature we are applying, suppose at 121 degrees Celsius for sterilization purpose. So instead of applying that, we can apply temperature less than 100 and pH we can maintain at low pH, low water activity. So because of the multiple hurdles, we can get the same level of the effect or in the killing of the microorganism, and also the fruits or the vegetables will be more fresh like. So we apply multiple preservative methods so that the multiple components or cellular components, they can be targeted at the same time, and the microorganism will not going to cope up with all these changes simultaneously. So we apply these hurdle technology. So here in this question, that because we are applying this hurdle technology, so the microorganism will be not there, and uh, food product can be stay good without refrigeration even, and we apply these different multiple preservative factors so that we can get the synergistic effect and we can kill these microorganisms, or we can even hamper their growth. So the assertion is true here and the reason is false. Okay, let's discuss now this question. So that read the following statements and identify whether they are true or false. So we have four statements about IMF, which is intermediate moisture food. So typically contains 10 to 20% of the moisture, and the water activity is between 0.75 to 0.85, and they are generally microbially unstable at room temperature, and they retain some sensory characteristics of fresh food products. We need to tell which statement of these four are, which statements are true and which statements are false. Okay, let's see the solution. So statement 2 and 4 are true. This is true. This is also true, and this is false. This is also false. So IMF, which is the intermediate moisture food, they are typically containing moisture around 20 to 50% weight by weight, not 10 to 20%. So statement 1 is false. And the water activity is having between 0.75 to 0.85, which is statement true, second is true. And they are microbiologically stable at room temperature. So statement 3 is false, and they retain some sensory characteristics like the color, texture, or the flavor of the fresh foods, which is true. So if we have a water activity level which is greater than 0.9, so they will be considered as high moisture food products like the cheese, sausages, and the candies. And if we have the like the product which is having moisture content from 0.75 to 0.85, they are the intermediate moisture foods like the syrups. And below maybe 0.6, the moisture, the food products are the below zero point 0.6 water activity. The food products are known as mostly the dry food products. So they are in the dry state. So we can see here in the dry food products, which is less than 0.6 water activity. So there is not any growth of the microorganisms or the yeast or the molds, but like and there is not much enzymatic reactions also take place. But between 0.7 to 0.85, there can be growth of this mold, yeast mainly, but the bacteria are they basically grow in high moisture food product, which is above 0.9 to 1. But some water activity, but some bacteria can even grow at 0.85 water activity as well. And then there are different other reactions like the enzymatic reactions or the non-enzymatic reactions. So they are at peak in this particular range. Or the hydrolytic reactions can also take place, and the lipid oxidation can also take place. So all based on this water activity level, we can say which kind of degradative, quality degradative reactions can take place in a food product. So based on the water activity level of a food product, its spoilage or its shelf life can be determined. So the unique features of that make IMF appeal to the consumers, it includes the conceived microbial safety, desirable odor, high nutritional value, and ready-to-eat products. So the purpose of making this IMFs are to achieve a desirable water activity by various ingredients so that the food products maintain enough water for the palatability and can be stored safely. Addition of the preservatives provides the margin of safety against the spoilage organisms like the Staphylococcus aureus is one of the microorganisms of the high concern which can tolerate water activity as low as 0.83 to 0.86 under the anaerobic condition. So we can sometime apply chemical preservatives in the IMS so that these kind of microorganisms can be avoided. So these are the different IMFs like papaya, pineapple, apple, banana, and the mango. So other examples of intermediate moisture foods include jams, jellies, candies, baked goods, honey, and semi-dried food fluid products. So these are basically mostly the semi-dried fluid products. So these IMFs and which is intermediate moisture food products, and the HMFPs, which is high moisture food products, they can be made like raw fruits can be taken, and the selection, washing, peeling, and cutting can be done. After that, washing and weighing can be done. Then blanching can be done. Uh, so 1 to 5 minutes in saturated steam, and then cooling in tap water or the cool, normal ambient water or little cold water. After blanching, soaking is there, which is in the addition of the sugar syrups and the additives. And after that, equilibration, which is the water activity around 0.93 to 0.98, and pH can be 3 to 4, and it can be stand for 5 to 10 days with gentle stirring at least twice a day. And then separation can be done. Okay, so this product can be basically self-stable high moisture food product, which is having the water activity 0.93 to 0.98 and pH 3 to 4. This is a high moisture food product, so it can be packed and it can be stored. But when we are making the intermediate food product, so separation process must be take place where the fruit can be separated from the sweetened juice. So sweetened juice can be packaged and stored, and this fruit which is in the which can be has been separated from the sweetened juice, it can be dried, and intermediate moisture food products can be obtained. It can be packaged and can be stored. So in this question, as we discussed that what are the intermediate food products? So they are basically the food products which is having water activity around or the moisture content around 20 to 50 and water activity around 0.75 to 0.85. And they are stable at room temperature and they are like fresh like products and they retain some characteristics, sensory characteristics like the fresh like products. Okay, so let's discuss now this question. In this question, column 1 is having food products like raisins, dried prunes, and then dried peaches and jams and honey. And in column 2, their water activity range is given. So we need to match the food products with their water activity level. Okay, let's see the solution. So B answer is the correct. Water activity level of the different food products is given, like raisins 0.512, 0.62, then dried prunes are the figs. They'll have the water activity 0.65 to 0.83. So raisins are having water activity is 0.51 to 6.62. Then prunes, they are having water activity of 0.65 to 8.3, 0.83. Then dried apricots or the peaches, they have the water activity of 0.73 to 0.81. So, then jams, jellies, and the marmalades, they are having water activity around 0.82 to 0.84. And last is the honey, which is having the lowest water activity or the very low water activity, which is 0.58 to 0.68. So here answer D is basically correct. So there are some other examples of the traditional intermediate moisture food products like the raisins and their water activity and the principal humectants which are using like the sucrose and the other sugars which are there already naturally present in the raisins, which is why the water activity of the raisins is 0.51 to 0.62. Then dried prunes and the figs, they are having water activity 0.65 to 0.83, and they are also having naturally having sugars or the sucrose, which is acting as a humectants. And then dried apricots or the peaches 0.73 to 0.81, and they are also having naturally occurring sugars or the sucrose as humectants. In the jams, jellies, marmalades, basically the water activity is 0.82 to 0.84, and the sugars or the sucrose are added uh, manually and not naturally present. In the sugared fruits, so water activity is 0.57 to 0.79. Sucrose and the other sugars can be added. Fruit fillings of the pastries, 0.65 to 0.71 water activity, and it can have sucrose and the other sugars. Then sweet chestnut puree 0.90, sucrose can be added as humectant, and in honey 0.58 to 0.68. So here in honey, sucrose can be there. Then there are fructose also and naturally present. In maple syrup, 0.82 to 0.86 is a water activity, and principal humectant can be sucrose or other sugars. And in chocolates, less than 0.55 is the water activity, and sucrose can be added as principal humectant. And in chutney sauces, NaCl and the food acids are the principal humectants, and the water activity is 0.86. So in these IMFs, having the different water activity ranges, so what kind of microorganisms can grow here? So if the water activity is little high, which is 0.8 to 0.84, so mostly the molds or the Saccharomyces species can be grown, where the foods like fruit juice concentrates or the chocolate syrups, maple and the fruit syrups and the fruit cakes. And if the water activity is 0.75 to 0.8, so halophilic or halophilic bacteria can be grown, or mycotoxigenic aspergilli can be grown, and the food products may include like jams, marmalades, marshmallows, or marzipans. And then water activity, if it's more low, like 0.65 to 0.75, so only the xerophilic molds can be grown. Saccharomyces bisporus can also grow, like the aspergillus species can also grow. So it includes marshmallows, jellies, molasses, raw cane sugars, and some dried fruits. And if the water activity is around 0.6 to 0.65, osmophilic yeasts like Saccharomyces roxy, or the molds like some species of aspergillus, they can grow in the dried fruits, which is containing 15 to 20% moisture, or some toffees or caramels or the honey. So here in this question, the different food products we matched already with their water activity levels. Okay, now we discussed that in these intermediate moisture food products, we have the humectants. So let's discuss some questions regarding humectants. So select the correct statement regarding the desirable properties of an effective humectant. Like it should be non-toxic, it should have high molecular weight, and it should show compatibility with organoleptic characteristics of the food. It should provide high caloric value, and it should reinforce the dietetic image by adding positive functional properties. So we need to tell which statement here are basically like correct here, and which is a desirable property of a humectant. Okay, so let's discuss here. So here, statement 1, 3, and 5 are correct. So humectants, they are basically the short-chained polyalcohols, so that prevent water losses and hardening in the products like toothpaste, and while also contributing to a creamy texture. Example includes like the glycerol and the sorbitol and the sugars. So they are the low molecular weight polyphenols or polyalcohols. So polyols also we say, and protein derivatives can also include, and minerals and organic salts can also act as humectants. So sometimes we see these kind of the nutrition bars or the breakfast bars, so they can also have this humectants, so that the texture or the shape of the bars can be maintained. So in the humectant, 3 hydrated waters can be present. So uh, with the humectant, closely related is the non-freezable water, which is a bound water, and then Intermediate water can also be there, so it can be in the bound or freezable form, and the free water is there, which is a freezable water. And this is the microstructure type of the water in the glycerine solution with various concentrations. So when there is a 0% glycerine is there, so there is a water which is free and in the bulk water. When there is this glycerine, so we can have the free water. This is the free water and this is the bulk water. And this water, which is a non-freezable water. So when there is some percentage of the glycerine we are adding, so some water which is in the direct contact with this glycerine, which is a humectant, that water will become non-freezable water. Other is a free water or bulk water. Then if 60% of the glycerine is added, most of the water will be non-freezable and little will be free water. And if 70% we are adding glycerine, so all the water is non-freezable water and 100% glycerine is this glycerine only. So an effective humectant, it should be basically non-toxic to ensure safety in the food applications, and they exhibit adequate solubility in the water and are of the low cost. It should exhibit compatibility with the organoleptic sensory characteristics of the food products, and the humectant should reinforce the dietetic image of the food by contributing positive functional property. Typically, this effective humectants, they are low to moderate molecular weight and uh, also have low caloric value. So here statement 2 and 4 are false, rest is true. So here this is a basically jerky product which is made from the spent hen meat, and they have this humectant like the glycerol. So control is this one, and when 10% glycerol is there, 15% and 10% sorbitol and 15% sorbitol is there. So we can see how the product is looking and how the texture is there. And here is the water activity level. We can see here in this image. So this is a sorption isotherm of jerky products, which is made from the spent hen meat treated with the humectants like the glycerols or the sorbitols. So we can see this is the control one, and this is the 10% glycerol, and this is a 15% glycerol. So when we are adding glycerol, so moisture is retaining more in the 15% glycerol when we are adding. So moisture retention is more in the jerky. So this way the humectant is basically acting. So here in this question, statement 1, 3, that it is a non-toxic. Humectants are non-toxic and they show compatibility with the organoleptic characteristics of the food, and they also it should reinforce the dietetic image by adding the positive functional properties. True. Okay, so let's solve some of the numerical problems now. So what will be the water activity of the sugar solution that contains 9 mole of the water and 1 mole of the solute? So water activity is basically the ratio between the vapor pressure of the food product itself, which is P, within a completely undisturbed balance with the surrounding air medium or media, and the water activity of the distilled water under identical condition, which is P naught. So water activity is P by P naught, which is vapor pressure of the food divided by vapor pressure of the distilled water. So in this question, we need to find the water activity of the sugar solution that contains 9 mole of the water and 1 mole of the solute. So when we are adding in a completely pure solvent, when we are adding some solute molecules, we usually follow the Raoult's law. So the ideal solution, it says that the ideal solution, the partial vapor pressure of each component in a solution is basically directly proportional to its mole fraction in the liquid phase, which is P equals to XP naught. So when we are adding some of the solute, or then the vapor pressure changes, because that will going to occupy the some of the space in the surface. So the partial vapor pressure also goes down when we are adding some of the solute. Okay, let's solve this question. So as we know that this is a pure water, which is reference. So it is having water activity 1. When we are adding sugar, like in the sugar solution, we have with the 9 moles of the water and 1 mole of the sugar. So they are making 10 moles. So it is having sugar also and water molecules also. So the mole fraction here in the water, which is equals to the water activity. So the water activity of an ideal solution is a direct function of the mole fraction of the solved components. So the water activity is basically equals to the mole fraction, which is equals to the moles of that particular component. So moles of water divided by the total moles, which is moles of the water plus moles of the solute. So mole fraction of water is equals to, basically this is a mole fraction of water is equals to the mole of the water, which is how much moles of the water is there, 9 moles, right? So we will keep 9 divided by the total moles, which is moles of the water plus moles of the solute, which is 9 plus 1, 10. So 9 divided by 10 is 0.9. So as we are adding the sugars or the solute, we are adding, so the mole fraction of the water is decreasing. Hence, water activity will also decreasing and it will become less than 1. And if it's a pure water, the water activity will be 1. So here, C is the correct answer. I hope this question is clear to everyone. Let's discuss this next question. The carrot slices having the water activity of 0.89 are to be preserved using osmodehydration, addition of the salt NaCl to 20% sugar solution, sucrose solution, which is having a water activity of 0.987, reduces the water activity to 0.85. Percentage of NaCl added to the solution is, and we the sucrose molecular mass and NaCl molecular mass are given. The carrot slices are there, which is having the water activity of 0.89. And it is to be preserved. So we have the solution, sucrose solution, 20% sucrose solution, which is having water activity 0.98, and we are adding some salt in this. So the water activity of the resultant water will be 0.85. And we need to tell how much NaCl we need to add here. Please try to solve this question. Okay, so let's understand the question. So we have this 20% sucrose solution initially, which had the water activity 0.987. So it is having 80% water and 20% sucrose in the solution. Then we have to add some NaCl of known percentage, which will going to decrease this water activity from 0.98 to 0.85. So this will become sucrose plus NaCl in water, right? So we know sucrose 20% is there. Then NaCl, we are considering X. We need to find out this, and water is there. And we will going to add this carrot slices, which is having water activity 0.89, so that to prevent the moisture loss from carrot slices during the osmodehydration, the solution should have water activity lower than 0.89, right? So find the percentage of NaCl which is going to add to this 20% sucrose solution so that the water activity will become this 0.85. And the molecular mass of sucrose is 342 gram per mole and molecular mass of NaCl is 58.44 gram per mole, which is given data in the question. Okay, so suppose the final solution is of 100 grams. So what NaCl we are adding is X gram. So the water in the final solution will be 100, which is the total solution, minus 20, which is the sucrose, minus X. X is the NaCl, right? So this here we are discussing in the final solution. This final solution is 100, total 100 grams, suppose. In this 20 is the sucrose and X is the NaCl. So 100 minus 20 minus X would be the water. So 100 minus 20 is 80 minus X gram is the, so 80 minus X gram is the water in the final solution. Now moles of the water if we want to calculate, so moles formula is mass divided by the molar mass, right? So mass of the water in the final solution is 80 minus X divided by its molar mass, which is, we all know that the molar mass of water is 18, how? H2O. O is 16 molar mass and hydrogen is 1. So 1 into 2 plus 16, which is 18. So 80 minus X divided by 18 is the moles of water in the final solution. Now moles of the sucrose would be 20% is the sucrose. So 20 divided by its molar mass. Molar mass is, molecular mass is 342, right? So 20 divided by 342 would be 0.05848. Now moles of NaCl would be X amount of the mole, X amount of the X gram of the NaCl is there in total solution or in the final solution, and its molar mass is 58.44. So X divided by 58.44. So we have this moles of water, sucrose and NaCl now. Since NaCl is basically dissociated into Na plus and Cl minus, so the effective moles of the particle would be multiplied by 2. So 2x, 2 into x divided by 58.44 is the actual moles of the NaCl, because it is going to dissociate in Na plus and Cl minus. So now applying the Raoult's law, so the water activity in the final solution, which is finally is 0.85, which is this, is equals to its mole fraction, which is equals to okay. So we were discussing this question, right? So water activity is equals to 0.85 of the final solution, which is this, is equals to mole fraction, which is XW, equals to the, this is for water. So the, what are the moles of the water divided by the total moles, right? So the, in the final solution, what is the moles of water, which we calculated here? Moles of the water is 80 minus X divided by 18. divide by total moles. Total moles are moles of water, which is 80 minus x divided by 18 plus the moles of sucrose, which is 20 divided by 342, which is 0.05848 plus the moles of the NaCl, which is 2x divided by 58.44. Now let's solve this. So this numerator will come here, which is 80 minus x divided by 18 is equals to, and the denominator will be multiplied by this 0.85 is equals to, so 0.85, and this denominator, which is 80 minus x divided by 18 plus 0.05848 plus 2x divided by 58.44. Now 80 minus x divided by 18 is kept same. Now multiply this 0.85 with the whole this total moles. So 0.85 into 80 is 68 minus 0.85x divided by 18. Second plus 0.85 multiplied by this 0.05848, it would be 0.049708 plus 0.85 multiplied by this 2, it would be 1.7x divided by 58.44. Now further this equation we are keeping, and we are solving now this equation. So it would be 80 divided by 18 minus x divided by 18. So we are basically separating here the numerical value with the terms which are having x value. So the 80 divided by 18 is separated, and minus x by 18 is separated, equals to 68 divided by 18 minus 0.85 x divided by 18, and this 0.049708 is kept same, plus 1.7 x divided by 58.44 is kept same. So yeah, so this from here what we are doing, 80 minus, we are taking this term 68 by 18, so 80 minus 68 divided by 18, it would be this 12 divided by 18 minus x by 18, which is this term. Then this term will be in plus, so it will be plus 0.85 x divided by 18. So it would be minus 1 plus 0.85, which is minus 0.15 x divided by 18 equals to, this is kept same, which is 0.049708 plus 1.7 x divided by 58.44. Now 12 divided by 18 is 0.6667 minus this term, which is 0.049708 equals to we have this term of x and this term of x, so it would be plus now, right? So we can keep this both and it will be in plus. So it would be 0.02909 x plus 0.00833 x. So this is basically when 0.15 we are dividing by 18, it would be 0.02909 x, and when we are dividing 1.7 x by 58.44, it would be 0.0833 into x. So this x would be equal to, basically when we subtract 0.6667 minus 0.049708, it would be 0.617 divided by this summation of this, which is 0.02909 x plus 0.00833, which would be 0.037. So x would be 0.617 divided by 0.037, which would be equal to 16.67 gram, or when we convert it, it will be 16.67 percentage. So the answer is, when we are in this solution, we are adding, basically in sucrose solution, we are adding this 16.67 percentage of the NaCl. I hope this question is clear to everyone. Let's move to this next question, that in the concentration of the orange juice. So we have this orange juice and we are concentrating it into an evaporator. So a fresh extracted and strained juice, which is having 7.08 weight% solids. So we have this orange juice, which is having 7.08% solids, is fed into this vacuum evaporator, and in the evaporator, water is removed. So W is representing the unknown amount of the water, and the solid content is increased. So this the concentrated juice amount is C, so which is having 58 weight% solids, and we are giving this speed 1000 kg per hours of the juice. Now we need to calculate the amount of the outlet streams of the concentrated juice and the water. So we need to calculate C and W value here. Please try to solve this question. Okay, let's solve this question. So when we are doing here the material balance, so it will be total inlet equals to total outlet. So total inlet is 1000 is equals to W plus this C. That is equation 1. And when we are doing component balance on the solids, it would be 1000 into 7.08%, which is 7.08 divided by 100, is equals to W into 0, because it is a water, it is having no solid content, so it is a 0% solids in this, plus C into 58% of the solids, so 58 divided by 100. So this equation would be 7, this 7.08 divided by 1000 into 100, 7.08 divided by 100 into 1000 would be 70.8, and equals to, this would be 0, and it would be 0.58. So 58 divided by 100 is 0.58 C. So C would be 70.8 divided by 0.58, right? So C is basically 122.07 kg per hour. Now we can substitute this value of C from equation 2 in equation 1. So W would be, so it would be 1000 equals to W plus C, which is 122.07. So W would be 1000 minus C, which is 122.07. So W would be 877.93 kg per hour. So this is the amount of the water we are getting, which is 877.93 kg per hour, and the concentrated juice is 122.07 kg per hour. I hope this question is clear to everyone. We have this next question, which is about the production of jam from crushed fruits in 2 stages. So in a process which is producing jam, C1, crushed fruits around 14% of weight soluble solids is mixed in a mixture with sugars around 1.22 kg sugar per 1 kg crushed fruits, and the pectin is around 0.0025 kg pectin per 1 kg of the crushed fruits. The resultant mixture is then evaporated in a kettle to produce basically jam, which is having 67 weight percent soluble solids. So for a feed of 1000 kg crushed fruits, we need to calculate kg of the mixtures from the mixer and kg of the water evaporated and the kg of the juice which is produced. So basically we have this mixture for mixing of these crushed fruits, which is around 14% soluble solids and 1000 kg. We are feeding with the sugar and with the pectin. And then there is some output will be from the mixture, which is further fed into evaporator for the evaporation in a kettle, and water is evaporated and the jam is produced, which is 67 weight percent of the soluble solids. So we need to calculate here what is the water evaporated and how much is the kg of the jam is produced and how much of this mixture was produced. Please try to calculate this question. Okay, let's solve this question. So the crushed fruits, we know 1000 kg is there, and the sugar is given as this 1.22 kg of the sugar per 1 kg of the crushed fruits, right? So when we are saying that the crushed fruits are of 1000 kg, so if for 1 kg of the crushed fruit, we need 1.22 kg sugar. So for 1000, we can multiply by 1000. So it would be 1.22 into 1000, which is 1220 kg of the sugars we are adding in the mixture. Then how much is the pectin we are adding? 0.0025 kg pectin in the 1 kg of the crushed fruit. So for 1000 kg of the crushed fruit, we can add 0.0025 into 1000, which is 2.5 kg of the pectin we are adding in the mixture. So the total weight of the input we are giving in the mixture is 1000 crushed fruits, then 1220 is the sugar and 2.5 is the pectin, which is equals to 2222.5 kg total weight of the input mixture. So this is the weight of the input mixture, 2222.5. Now, soluble solids which are entering in the process, we can see this. So the soluble solids which are from the crushed fruit is around this 14 weight percent. So 14 divided by 100 into 1000 is 140 kg is the soluble solids from the crushed fruits. Now, sugar is the completely soluble solid itself. So the total sugar, which is 1220, is all that soluble solid, and the pectin is also treated as soluble solid. So 2.5 kg is the soluble solid from the pectin. So the total soluble solid would be 140 plus 1220 plus 25, which is 1362.5 kg from the sugars, from the pectin, and from the crushed fruits. So when the evaporation is taking place, only water is removed. Therefore, this soluble solid, which is 1362.5 kg of the soluble solid, it will remain in the jam. So the jam which is produced is having here given as 67 weight percent of the soluble solid. So 67 divided by 100 into jam, which is into J. J is the mass of the jam, is equals to this value 1362.5 kg, because it is a total soluble solid. So the total soluble solid will be same which is in the mixture as the same in the final product, because only water is remaining, right? So J would be here 1362.5 divided by 0.67, which is 2033.6 kg is the jam which is produced as a final product. Now, what is the amount of the water evaporated? It is weight of the total input mixture minus weight of the jam which is produced. So it is this 2.5 minus 20.33. So their subtraction will give the water evaporated, which is 188.9 or 189 kg of the water is evaporated. I hope this question is clear to everyone. So here this water is around 189 kg. This mixture is around 2, 2, 2, 2.5, and the jam which is produced is 2033.6. Okay, let's see next question. So a membrane separation system is used to concentrate the total solids in a liquid food. So there is a liquid food and it is having 10% total solids, and we are using a membrane separation system to concentrate this liquid feed to 30% total solids. The concentration is accomplished basically in two stages, with the first stage resulting in release of a low total solids liquid stream. So first we are using membrane for this liquid, and then if we are getting this 25% TS, so after that we are again using the membrane and we are getting 30% TS. But if we are not getting, that is recycled and water is removed in this process, right? So in the second stage, it separates the final concentration product from a low total solid stream, which is returned to the first stage. So this is returning to the first stage. Determine the magnitude of the recycled stream when the recycle contains 2% TS. The waste stream contains 0.5%. This is the waste. And the stream stage between 1 and 2 contains 25% of the TS. And the process should produce around 100 kg per minute of the 30% TS. So basically there is a liquid which is fed, which is having 10% TS, is fed in this first membrane separation. So the waste is removed as 0.5% TS, and the remaining will go, which is having 25% TS, will go here in the second stage. So if 30% TS we are achieving, we can say it is a final product and it is having 100 kg per minute production. But if we are not getting, so the 2% TS stream is getting recycled and it will come in the first stage again, and then again it will go in the second stage as a loop or as a recycling thing. So here in this, we need to basically calculate the magnitude of the recycled stream. Okay, solve this question. So let's see the solution. So the material balance in the total system, this total system, if we talk about the material balance, would be F is equals to W plus P. So F is equals to W plus P, where P is 100. So the P is basically 100 kg per minute, right? So F equals to 100 plus W is the first equation. Now the component balance in the total system would be F into its fraction XF equals to P into XP plus W into XW. So X is the basically solid fraction which we are having for each component, for feed, for waste, and for product. So F into, since we know that the solid fraction is 10% for the feed, so F into 10 divided by 100 is equals to, in the product, we know 30% is the TS. So 100 into 30 divided by 100 plus in the waste, it's 0.5% TS. So 0.5 divided by 100. Now it would be 0.1 F equals to 30 plus 0.005 W is the second equation. Now in the case of first stage, now this is the first stage. Let's discuss here component balance. So F plus R equals to W plus B. So material balance is F plus R equals to W plus B, and the component balance would be F into XF plus R into XR equals to W into XW plus B into XB. So as we know that feed is a 10%, having 10% TS. So F into 10 divided by 100 plus in recycled stream TS is 2%. So R into 2 divided by 100 equals to, in waste it's 0.5. So W into 0.5 divided by 100, and B is 25% TS. So B into 25 by 100. So this is equation 4. We have made 4 equations from the material and component balance, from the total system and the first stage in regards to the first stage. So now put value of W, or sorry, value of F, which is F equals to 100 plus W, in the equation, this one, 2. So it would be 0.1 into F. So 0.1 into 100 plus W equals to 30 plus 0.05 W, which is this. So when we solve, it would be 0.1 into 100 would be 10. So 10 plus 0.1 W equals to 30 plus 0.005 W. So W would be 10 or 30 minus 10, which is 20, divided by 0.1 minus 0.005, which is 0.095. So W would be 20 divided by 0.095, which is W is 210.526 kg per minute. So here we calculated W. So F would be 100 plus W. So 100 plus W, which is 100 plus 210.526. So F would be 310.526 kg per minute. Now we have value of F and W. So we can put into this equation 3. F plus R equals to W plus B. So F, which is 310.526 plus R equals to W, which is 210.526 plus B. So this equation 5. So this equation will become R plus, R plus 310.526 minus 210.526. So it is 100. So 100 plus R equals to B is the equation 5th. Also we can put value of F and W in equation 4, which is this equation. Right? So F divided by F into 10 by 100 plus R into 2 by 100 equals to W into 0.5 divided by 100 plus B into 25 by 100. So F would be here 310.526 into 10 by 100 is 0.1 plus R into 2 by 100 is 0.02 equals to W is 210.526 into 0.5 divided by 100, which is 0.005 plus B into 25 divided by 100, which is 0.25. Now we can put the value of B from here, which is 100 plus R. Right? So it would, this equation would be 310.526 into 0.1, which is 31.0526 plus 0.02 R equals to 2210.526 into 0.005, which would be 1.05263 plus B, plus 0.25 B, and we are putting B as 100 plus R in this next equation. Rest would be same. So now what we can do is 31.0526 0526 plus 0.002 R equals to 1.05263 plus now 0.25 into 100 is 25 plus 0.25 into R is 0.25 R. Now we can separate this numerical values, which is 31.0526 minus this 1.05263 minus this 25 equals to this R value, which is 0.25 R minus this 0.02 R. So 31.0526 minus 1.05263 minus 25 would be 4.9997 divided by 0.25 minus 0.02 R, which is 0.23. So R would be here R is equals to this. So R would be 4.99997 divided by this 0.23. It would be 21.739 kg per minute is the R value, which is recycled stream value, this value. I hope this question is clear. Let's move to the next question, which is about high moisture food products. So which of the following statements regarding preservation of high moisture food products are incorrect? So blanching helps in inactivating enzymes and contributes to microbial stability. Second is lowering the pH increases the microbial activity in high moisture food products. And third is water activity depression is an effective hurdle to limit microbial growth. And fourth is addition of sulfides act as an anti-browning agent in high moisture food products preservation. We need to tell which statement is incorrect. Okay, let's discuss this solution. So only second statement is incorrect. So high moisture food products, they are typically the one which is having a high water activity as high as 0.98 and can stabilize using the help of different hurdles or the combination of different hurdles. So pH exert a strong selective pressure on the existing microflora. So pH range is around 3 to 4.1, and it will help in the control of the microbial growth. So it might also not represent the optimum for growth. So because of the low pH, the microorganisms will not grow. pH water activity interaction in the applied range will be enough to suppress the growth of the most of the bacteria of the concern in the food preservation. At high levels of water activity, the effective pH on osmophilic yeast might be the same as for non-osmophilic yeast. So the preservation methods used for the HMFPs are common hurdles used for providing the microbial stability to the HMFP products are blanching, water activity depression, lowering of the pH, addition of chemical preservatives like adding of the sorbates or the benzoates, and the addition of the sulfides as an anti-browning agent. In the blanching is basically commonly used as a harder technology so that to destroy the enzymes that could lead to the spoilage, and it also reduces the microbial load and helping in the microbial stabilization of high moisture food products. Then lowering the pH actually reduces the microbial activity because most of the spoilage and the pathogenic microorganisms cannot thrive in acidic environments. Then reducing water activity limits the availability of the free water, which is essential for the microbial growth. So hence it is a critical hurdle for high moisture food products preservation. And then the sulfites can be used to inhibit the enzymatic browning reactions by the polyphenol oxidase, and also it can have the antimicrobial properties. This makes them valuable in preserving the color and extending the shelf life. So this HMFP technology is basically successfully applied to peaches, peach halves, then pineapple slices, mango slices, and purees, and also the papaya slices, and also the chico zaperte slices, then banana purees, plum, and then passion fruits, tamarinds, whole figs, and then strawberries and formalica. So this methodology applied or employed was based on the combination of basically the mild heat treatment such as blanching for 1 to 3 minutes with saturated steam. Slightly we can reduce the water activity like 0.98 to 0.93, and by addition of this sucrose basically, or also the glucose, and also we can lower the pH like 4.1 to 3, and we can add basically the citric acid or the phosphoric acid to lower the pH, and we can also add antimicrobial agents which are like 1000 ppm of potassium sorbate or sodium benzoate, as well as we can add 150 ppm of the sodium sulfites and sodium bisulfites to the product syrups. And during the storage of high moisture food products, the sorbates and the sulfite levels they decreased, as well as the water activity levels, and due to the hydrolysis of the glucose. So we can see here that there are these different different food products, and we can use these different combined methods for the minimal processing of the uh, for the preservation, and here the temperature, storage temperature, and the shelf life. So in the peach sliced halves or the whole, blanching for 2 minutes using the steam, and water activity around 0.98 using the sucrose, pH 3.7, and this sodium sulfites, sodium hydrogen sulfides, they are 150 ppm, or this Ks, which is potassium sorbate, 1000 ppm is added, and we can store the product at around 35 degrees Celsius for around 3 months. Same as like different food products are there, same for different food products basically are there. So suppose in the we are saying this for strawberry. So for strawberry we can do blanching for 1 minute, then water activity we can keep 0.97 for with sucrose and 0.95 with glucose, and pH 3.1, and ascorbic acid around 200 ppm. Then sodium hydrogen sulfides 150 ppm, and the potassium sorbate around 1000 ppm. And we can keep these strawberries for around like 25 degrees Celsius even for 4 months. So like these for papayas, for mangoes, for pineapple, sliced or whole papaya, and then peaches for or like the half peaches. We have the different combined methods given, and we can put all these together with together, and that is a combined or a hurdle technology approach, and we can keep the fruits or these fruits at or near ambient temperature around 25 to 30 or 35 degrees Celsius, and for a period of 3 to 4 or 5 months we can keep using these combined hurdles. And we can see these hurdles are not very strong hurdles, so we are just using them in the like mild treatments we are giving, and because of the combination of these hurdles, the mild hurdles only, and the fruit quality can be retained properly, and uh, simultaneously we can achieve a good shelf life at the normal temperature itself. So this is the preparation of the self-stable high moisture fruit product. So we can select the fruits, wash, peel and cut, and then blanching is done with the saturated steam for 2 to 3 minutes, and water cooling is done. And after that, the product equilibration can be done, like the addition of the sugar, acids, potassium sorbate, sodium bisulfites, or sometimes the wet or dry infusion at atmospheric pressure can be done. We can achieve the water activity around 0.94 to 0.98, and pH around 3 to 4.1, and then 400 to 1000 ppm of the potassium sorbate and 150 ppm of the sodium bisulfite. So these fruit pieces, they can be basically poured into the glass jars or polythene bags and covered with the uh, basically uh, some syrup, and then it can be packaged and sold. So here in this question, we need to tell which are the incorrect statements. So the blanching, basically it helps in inactivating the enzymes, which is true, and it also contributes to microbial stability. So it is true. And lowering the pH basically increases the microbial activity, which is wrong, because when we are lowering the pH, the most of the microorganisms will not able to thrive. Then water activity depression is an effective hurdle to limit microbial growth, which is true. And then addition of the sulfides, it acts as an anti-browning agent in high moisture fruit products, so which is also true. So only statement 2 is incorrect. Rest all 1, 3 and 4 are true. Okay, now let's discuss about the fresh cut produce, which is the last topic. So the fresh cut produce, they are basically any fruits or vegetables or the combinations when that has been basically physically altered from its original form, but it remains in a fresh state, is the fresh cut produce. So fresh cut produce may consist of the like the peeled, sliced, shredded or trimmed or washed fruits and vegetables. Yeah, so yeah, so washed fruits and vegetables. So these are the minimally processed, just we are washing or we are just peeling or slicing, shredding. We are just removing the peel, or we are just converting into the cubes or sliced form, or we are trimming. These are the cut fruits or the cut fresh produce. Basically, we are not giving any kind of treatment to this kind of food product. So first step in this making the this fresh cut produces is first we receive the any fruit or vegetable, and we do the inspection, and the pre-cooling is done, and cleaning, washing and disinfection is being done. After that, sorting and grading is done, and then peeling, core removal and cutting is done, then washing and the sanitation is done, which is a kind of the hurdle technology, and the packaging is another hurdle technology in this. So, and we can also use some kind of other hurdles like dipping treatments or the thermal treatments and the other treatments in the step 4, uh, or after step 4 or between step 5. So this kind, like the minimal, we are processing these fresh cut produce and we are packaging and we are selling. That is a fresh cut produce. So let's discuss one question regarding this. So column 1 is having different pathogens like the Clostridium botulinum, Salmonella species, and Listeria monocytogenes. And in column 2, we have the different commonly, like in which cut produce, fresh cut produce, it is commonly found. These kind of pathogens in fresh cut produce, in which fresh cut produce, which pathogen is found. Okay, let's discuss the solution. So first is true. So basically, um, this Clostridium botulinum is found in the shredded cabbage, cubed butternut, squash, and the sliced onions. And the Salmonella species, it is found in chopped or sliced tomatoes, beans, sprouts. And then Listeria monocytogenes, it is found in cabbage salads, broccoli, cauliflower, and coleslaw. Coleslaw. Okay. So the quality changes in the fresh cut produce may includes like the appearance may change, browning can be there, texture change means softening can be there, flavors can change, so the foul smell can be generated, or the nutritional or health related or the nutritional value may decrease. Also the safety aspect like the microorganism may grow, the pathogen introduction can be there regarding the when the quality is changing in any fresh cut produce. So the fresh cut produce and the vegetable, they are basically susceptible to the microbial attack due to the processing steps used for the preparation of its, like the peeling, cutting, slicing, etc. So the Clostridium botulinum is found associated with shredded cabbage, cube, butternut, squash, sliced onions. So these are the conditions prone to anaerobic growth where spores may survive. Salmonella species is commonly linked to the chopped tomatoes and bean sprouts due to their high water activity and contamination potential. And Listeria monocytogenes is found in refrigerated ready-to-eat salads like cabbage salads, then cauliflower, coleslaw, and because it thrives or survives in the low temperature. And if we see Staphylococcus aureus, it survives basically in the ready-to-eat vegetable salads. Then parasites like Cryptosporidium species is found in basically lettuce or the onions. Then Shigella, it can be found in the shredded lettuce, green onions, shredded cabbage, chopped parsley, and E. coli O157H7 can survive in lettuce, sprouts, mixed vegetables, cilantro, coriander, celery, etc. So the quality changes of these fresh cut fruits and vegetables, it includes like they are the different commodities like apple, beans, strawberries like that, and then they are in the their cut form. So these are the main degradation can happen like the browning or the yellowing or the surface drying, and the beneficial modified atmosphere can be given to them, like percentage of CO2 can be altered and the O2 percentage can be altered like this. Like sliced apples, they will go brown. So less than 1% of the CO2 atmosphere can O2 atmosphere can be given. And if we see in the like strawberries, so sliced or topped strawberries, they lose the texture and the juice and the color. So 1 to 2% O2 and 5 to 10% of CO2 atmosphere can be provided to it. Same for the other fruits, like for oranges, if we see, so sliced or the sectioned oranges, they can have the leakage. So 14 to 21% of the O2 and 7 to 10% of the CO2 atmosphere can be beneficial for them. So here in the question, answer A is correct here. Okay, so the topics we covered today is mainly the minimal processing of fruits and vegetables. So it includes about basically what are the different key requirements or the operational steps, and what are the different quality changes occur, or what are the different safety issues can be there. Or then we discussed about the hurdle technology concept, its type, and how different hurdles will going to affect the microorganism, and how they are going to like their mechanism, like the disturbance of the hemostasis or multi-target approach, or exhaustion of their metabolic energy, and the stress shock proteins, like all these mechanisms we discussed. And then we discussed about the effect of the hurdle technology on the product quality. Then we discussed about intermediate, and we discussed about some numericals, and then we discussed about the intermediate and high moisture fruit products, the preparation and how they're processed and the preserved. Then we discussed about the cut fruits and vegetables and their manufacturing process, like quality changes, and we discussed about the different anti-browning, like the different chemicals or other things or the packaging we can use to enhance their shelf life. In the next class, we'll be discussing about juice extraction and clarification, concentrates and pastes, then aseptic processing and packaging, and RTS and RTD beverages, and quality and safety aspects. So I hope there is no doubt in the today's class. So I'll be providing the lecture recording on my YouTube channel, and the videos I'm providing regularly for every week. And this is the basically where you can find the PPT of the lecture, and you can ask your questions on Q&A discussion forum. So thank you so much for joining the class. Also, please submit your feedbacks. When you get the feedback link, please try to submit the feedback, and please let me know if that my way of teaching is correct, and if you want any other modification. So thank you so much for joining the class today, or listening the recorded lecture videos also. So this is the end of this today's session. I'm stopping the recording.